throughout the entire chapter on microbial genetics, we were talking about what? The problem? You couldn't find? Yeah, you good? That is, I know it is a simpler part. I know that when these words come out of my mouth, it's not really reassuring, but now when we talked about the gene expression, what? Yeah, sure. When we talked about gene expression, we mentioned that expression is regulated. There are many different ways how to regulate the expression, but we're going to talk about two major ones. One, the Operon is classic way. Riboswitches were discovered relatively recently, okay? And they pretty much control whether genes, certain genes, being expressed, which means whether genes are transcribed or translated. Does that make sense? What I just said, okay? So now, first we're going to talk about the Operon. A gene, the result of the gene expression, what it is. What is the result of the gene expression? The end result. Protein. Yeah, it's protein. Does that make sense? In bacteria, genes are often organized in operons, which means that proteins that altogether confer a certain trait, perform a certain function, grouped together. Think about the construction team, like people that do roofing, okay? You don't hire one guy here, one guy there. Usually what happens, you call to the company and they send the entire team of people and each of them knows his specialty and they do the roofing, okay? So it's a team. Operons are teams of genes. Does that make sense? Operon consists of so-called structural genes that actually encode for proteins that perform certain function. Function can be to produce a specific protein or to metabolize a sugar, right? Upstream of the structural genes is so-called operator sequence that regulates whether structural genes are going to be expressed or not. And upstream of operator is promoter, the site for recognition by RNA polymerase, right? Because that's where transcription starts. Now, the role of operator, I can compare to the situation when, so imagine that you are RNA polymerase. You're like a runner at the start line. If nothing, if there are no obstacles, no walls, you can run to the finish line. So you can do the transcription. But if somebody puts the wall in front of you, some kind of a wall here on the operator sequence, transcription cannot occur. You cannot run. Does that make sense? Okay. Sometimes regulatory genes upstream of promoter are also involved in the regulating the expression of the structural genes of the operon. Is that more or less clear, the structure of the operon? Structural genes do the job, promoter initiates the transcription, and operator decides, regulates whether the transcription will go or not. And sometimes regulatory genes get involved. Now all these structures, promoter, operon, genes, in which nucleic acid you can find them? Say it, come on, no points. I'm not going to punish you for it. In which nucleic acid are you going to find them? genes, DNA. It's DNA, okay? It's a chromosome. It's a DNA, okay? Does that make sense? The fundamentals. Now, riboswitches are the structures in the mRNA. This looks awfully complicated. Now, can you think of any switch that you know? Just switch. What? Bingo, light switch. How does it work? You flick it on or flick it off. You just explained how riboswitches work. Seriously, if you ever disassemble the light switch, I'm not an electrician by the way, but just there's some little thingies inside of it that go like up and down, and they touch this contact, touch that contact. So it's kind of a lot of mechanics. It pretty much every time you flick it on, something inside changes shape. Does that make sense? You flick it off, something changes shape. Does that make sense? That's light switch. Now, riboswitch. It works conceptually the same way. This is the special sequence of mRNA that forms the stem loop structure. Downstream of the riboswitch is another stem loop that is called anti-terminator. That anti-terminator sequence allows mRNA to continue transcription. As long as anti-terminator is present, RNA polymerase will go on and transcribe DNA. You see DNA here? The pale blue line? Does that make sense? If the small molecule, some kind of organic molecule, for example, will bind to the riboswitch, that loop structure in the riboswitch, the mRNA shape changes. You see that? And that converts anti-terminator loop into the terminator loop. And due to the structural interactions between the mRNA and RNA polymerase, when terminator loop appears, transcription stops. Does that make sense? Yes.
I guess I don't understand why mRNA and DNA are on the same string.
Say it again.
Yeah, so why, how do you have both mRNA, mitochondrial RNA, and DNA?
Oh, it's not mitochondrial, it's messenger. So, during the transcription, DNA is transcribed into mRNA. So this guy here, sorry, let me use different color. mRNA is synthesized continuously. And RNA polymerase reads DNA. Does that make sense so far? That's transcription.
Okay, so the mRNA that is there, that is created by the RNA polymerase?
This one?
Yeah.
Yes. Yes. So as long as the structure is maintained, as long as the structure of mRNA is maintained, transcription is going to proceed. When structure is changed by some kind of a signaling molecule, RNA polymerase is going to fall off DNA. It's a bit more complicated because what can serve as the regulator? What this molecule, what can it be? It can be, for instance, some quorum sensing molecule, the one that is produced by other bacteria, the one that tells everybody in the population, look, there's too many of us. Stop making this gene. Stop making that gene. Does that make sense? Okay? Conceptually, what you have to understand is that in case of RIboswitch, the small molecule regulator, some kind of organic molecule, binds to the RIboswitch and shuts down the transcription. But RIboswitch is not on the DNA. RIboswitch is on the RNA. So it regulates the process of transcription and the process of translation, which works exactly the same way. Okay, which we'll chat in a second, okay? By binding to RNA but not DNA. Does it make sense? Sure. You have mRNA that is made during transcription. Part of that mRNA is called the riboswitch. Does that make sense? So far? Okay. If the chemical, the molecule, binds to the riboswitch and changes its shape, the changed shape is the signal for transcription to stop. Does it make sense? That's it. That's all riboswitch. So from on to off. But the point is, the regulatory sequence, the riboswitch, the actual structure, is the part of mRNA. Okay? During the translation, similar things happen. You have small molecule that binds to the riboswitch and prevents ribosome from binding to mRNA. No translation occurs. Chemicals. It's the interaction between the small molecule and the RNA. RNA, its unique feature is that it can form many those stem loops. Okay? And through the interaction with ions, organic molecules, inorganic molecules, those stem loops can alter their shape. Okay? They can, instead of this, you're going to get this. Does it make sense? Seriously. Conceptually, what I want you to understand is that it's a switch. Riboswitch is the mechanism to turn on and off transcription and translation. It can turn off transcription when small molecule binds to special mRNA element and RNA polymerase falls off. And it turns off translation when small molecule binds to the mRNA sequence here, the riboswitch, and prevents ribosome from binding. Okay? No chemicals, everything goes smooth. You add chemicals, it shuts down transcription and translation by interaction with mRNA. Does it make sense? Yes.
So, graph A, what you have is you have the mRNA falling off, but graph B, you have the stopping of
Ribosome. Say it.
So in B, you have it stopping
In the right one.
polymerase.
No, it's a ribosome. Yeah, let me transcription, and this is translation. Does that make sense? So here it affects RNA polymerase, its ability to proceed, its ability to carry out the transcription. Here it affects the ribosome, its ability to carry out the translation. Right? operons. That is, if you're familiar with negative feedback, it's going to be very easy to understand. Remember that we have a promoter sequence, the operator sequence, and the set of structural genes. Let's talk about lactose operon. In the lactose operon, structural genes encode for the set of enzymes that can metabolize lactose. Does that make sense? These genes encode for enzymes that allow cell to use lactose as the source of energy and or nutrients. Without the expression of those genes, cell cannot use lactose. When they are expressed, cell can. That's the very first statement. Does that make sense? When genes are not expressed, cell cannot do anything about lactose. When they are expressed, cell uses lactose as a source, as a nutritional source. Okay? When lactose isn't present in the environment, the molecule of repressor protein is bound to the operator. Repressor blocks the transcription, physically blocks the transcription. So RNA polymerase cannot proceed. Is it clear? It's a wall. Go ahead. Is not there. Say again. Wait a minute. When lactose appears in the environment, it binds to the repressor, and the repressor dissociates from the operator sequence. Repressor is not blocking transcription anymore, and these genes now can be expressed. When these genes are expressed, I just told you which trait do they give a microbe. Able to use lactose. Let's write a chemical reaction. Lactose is used for whatever, and those lac genes, let's call them lac enzymes, okay? Enzymes. They metabolize lactose. Does that make sense? So lactose allows for the production of the enzymes that metabolize it. When lactose gets metabolized, what happens to its levels? They decrease. When all lactose is used, what's going to happen to this complex? Yes, there is no lactose. Repressor will go back to the operator and block synthesis of those genes. Does that make sense? Think about the winter. You hire the guys that clean up your driveway only in the winter, because that's when the snow is. When there's no snow, you don't hire those guys. Does that make sense? Or you see the trucks that clean the roads only when there is snow. And once everything is clean, they leave the roads. Does that make sense? So in this case, the substrate of enzymatic reaction induce the operon. Does that make sense? That's why lactose operon is called, do I have this word here? Inducible. So inducible operons are controlled via the substrate of enzymatic reaction. Does that make sense? When substrate is high, operon is induced, the products of the gene expression reduce the amount of substrate, operon shuts down. Now, what about operons that are controlled by the product? We're going to use tryptophan operon as an example. When there is no tryptophan, RNA polymerase can and will transcribe the genes responsible for its synthesis. When levels of tryptophan will rise, it will bind to the repressor. Repressor with the tryptophan bound will attach to the operator and stop expression of the genes. When expression of the genes stopped, what's going to happen to the tryptophan synthesis? It will stop. When the cell will use all tryptophan that was synthesized, what's going to happen to this complex? Yes, repressor is going to fall off and the synthesis will start again. So, products regulate repressible operons. Like any production facility, like when a company makes, I don't know, cars, and it made so many cars that everybody bought it, it's going to stop making these cars because there's no market anymore. Does that make sense? So, repressible operons are controlled by the product, inducible by the substrate. Make sense? Great question. No. Really great question. RiboSwitch is on mRNA. RiboSwitch is the next level of control. Let me do what I've done already. Just 1 second. So if this is DNA, okay, and this is Operon, okay, it can be controlled via this operator sequence, just as we discussed. Is that clear? That control is via DNA, okay? So operon may or may not, the structural genes here may or may not be transcribed. Does it make sense? Okay, I'm getting to the answer. They may or may not be transcribed, and it is controlled by this operator sequence. When they are transcribed into mRNA, this is where riboswitches are. Riboswitch, okay? Operon. Riboswitches, yes. Now, riboswitch it adds another level of control to the transcription, because although the operon here may allow, the operator may let transcription go, riboswitch adds another level of control. It can either shut down the transcription or prevent translation from happening. Does it make sense? So bacterial cell doesn't rely only on the operons to regulate the expression of the genes. It's also riboswitches. So those are independent from each other. different levels that control genetic expression. Does it make sense? We've also, just to remind you, we also remember we talked about histone-like proteins in the bacteria. The chromosomal DNA is wrapped around them in the bacterial cells. That's another level of control. If DNA is wrapped around histone-like proteins, genes cannot be expressed. Okay? So by controlling the shape of the chromosomal DNA, you can also control this. So if every cell is multiple levels that control genetic expression. Does that make sense? Okay.